Well, good morning, everybody. So I'm really happy to welcome you all to the Department of Botany webinar series. So this is a webinar for the month of April. We have with us Dr. Jasmine M. Shah. She's Assistant Professor at Central University of Kerala in Kasaragod in Kerala. And Dr. Jasmine is also working as a Joint Director of the Kasaragod Botanical Garden and Research Center maintained by the Central University of Kerala. And in 2012, she won prestigious DST Inspire Faculty Award. And among many distinctions of Dr. Jasmine, she is an executive committee member of the Society of Cytologists and Geneticists. And Dr. Jasmine holds her PhD from Madurai Kamaraj University in Tamil Nadu and postdoctorate from Indian Institute of Technology in Madras, IIT Madras. So I'm really happy to have you with us, Dr. Jasmine. And here, Dr. Jasmine is now going to talk on transfer of stress experiences in plants and their progeny. I met you first, Dr. Jasmine. I still remember way back in 2011 in the DST Inspire Faculty Award. So we both made it that time. The first batch of the faculty award of the DST Inspire. So yeah, over to you. I'm really happy to have it. Yeah, by the way, this particular research has been recently published in a very prestigious journal, and it has got a lot of media attention all over India. Many of the famous national dailies published it, the report, and it's about the epigenetics. The kind of molecular biology work that I don't do, but my colleagues always does it here. They're really passionate. Dr. Pankaj is here with us. I guess Sanjeev and Vinay is also joining. I guess they like this kind of studies. So I'm also excited to hear your story. Yeah, over to you, Dr. Jasmine.
Thank you so much, Felix. Firstly, for inviting me for this talk at Central University of Punjab. And it's so nice that you're arranging frequent seminars which will actually benefit students across the universities and research institutes. And yeah, you're an inspiration, Felix. I think we started our career almost at the same time, winning the same award. Yeah, as you said, I also was recollecting those days, in 2011 at Delhi. Congratulations, now you're associate professor. That's good. Soon I can see you going, reaching heights. And you're a very popular person in our country. Everybody, I think, knows Felix. When we say it, Central University of Punjab, that's the name which people in biological sciences think of. So there's one more thing which I want to share with you. These days, whenever I listen to Antarctica on TV or somewhere, apparently your name comes in the background of my mind. That's a great achievement, Felix. You have left your imprint in minds of people in science in our country and abroad. So
yes, I'm more into popularization, not really into the hardcore research like you are doing. Fantastic. I mean, you're doing great kind of research. I have one request. Could you please make it a full screen? Yeah.
Full screen, of my PPT?
Yes.
Okay, so let me start. So how much time do I have? Till what?
You take your time, no problem. Okay, so it's mainly for 1 hour, but if you can finish a little bit early, so we can have a couple of questions. So we have all the students here with us.
Okay, fine. So let's come to the presentation. So as you all know, I'll be talking about. One second. I was just trying to move away this. Transfer of stress experiences in plants to progeny, which is kind of triggered, memory of me in Felix, that topic. Otherwise, I think you would have forgotten about my existence. So, well, I was joking. Coming to this, As the name indicates, actually my slide is not moving. Can I just exit and come back? I mean,
yeah, sure. You can try again.
yeah. So before going into the technical details of this work, I will just share with you an experience with one of my school friends. So this WhatsApp and Facebook and all this has kind of reconnected our generation to uh, friends who have forgotten long back from school. And apparently, I met um, a friend of mine who is into economic, who's an economist and stuff like that, which I'm not familiar with, who stays in Bombay, who has no connection with science. But out of curiosity, one day he asked me like, what kind of research you do? And I started describing epigenetics in the most layman language possible to him. But after listening to me, he shared on WhatsApp this book, a page of this book, Sound Machine, just a short story written by Ronald Dahl, which has a collection of short stories. And he says that my research actually sounds to him, or he is reminded of this story when he thinks about epigenetics. So that's what a layman would think about epigenetics. So this story is nothing but it's about a scientist who, or a person, in fact, who has kind of, who is into discovering a machine which will capture sounds which are not audible to us, which are of higher or lower frequencies, and convert that the pitch of that sound into a sound which is audible to human ears. And he starts collecting sounds from plants. Like he identifies that when he cuts rose from his garden, or when he cuts a tree, he can hear the sound of the plants crying, which is at a different pitch, which is not audible to us, but with his machine, he's able to hear these sounds from the plants. So that's what memory in plants is like for people who are not into science. Well, coming to my topic, like I know the crowd here is all from biological science background, and you will be able to understand the technical side of epigenetics. So with that in mind, let me go into a little more deeper areas of epigenetics. So with respect to plants, actually, I'm a plant biologist, so my talk will be always, surrounding in and around plant. But epigenetic memory is seen in all eukaryotes, animals, humans. But I'm not going into that. So coming to plants, plants are a little different from other higher eukaryotes in the sense that they don't have locomotion. So they are forced to face the stresses which are there in the environment, right? So like whether it is a sunny day or rainy day, or if there is any radiation, they cannot run away. So that is the reason why they are kind of evolved to have a better adaptation in the environment to face unfavorable conditions in the environment. So we know there are biotic and abiotic stresses that plants come across, and these dynamic situations have forced plants to develop a dynamic adaptive strategy, that is, which is reversible, so that they can keep switching accordingly rather than running away from a stress. And that's how plants are able to survive in any kind of situation. Now, what we have been, if you look into the literature for past 30, 40 years into plant stress biology, whether related to biotic or abiotic stresses, in simplified term, it would convey the message that plants, they trigger a kind of signal like osmotic and oxidative stresses and disruption of osmotic and ionic homeostasis, and certain proteins will be expressed, transcription factors. There will be cell signal transmission, kinases would be playing their role, and so on. And a lot of chaotic things happening in the plants. Many different cycles are triggered, and finally gene expression happens. Expression for defense, expression for stress tolerance, and so on. So there are a huge number of papers unraveling the secrets which are happening in the plants in this area. And now what we are going to deal with in this presentation or in this session is whether these plants can remember these stress. So there are a lot of activities going on in the plants, but will they be able to reserve this memory of their experiences for a later time of their life cycle, or maybe even to the next generation? So we'll be trying to ask this question and finding answers for that. So before going into the molecular aspect, let me take you to a very simple experience. I don't know, like if you have observed nature, my teacher used to say, a good botanist is should have a good observation skill. They should be able to observe nature at a very closer level. So that's what I have been groomed right from my school days. And many times, especially in Kerala, we can get this feeling. We have very strong monsoons, June, July, August, sometimes it goes on even till September. So 2-3 months continuous rain, there will not be absolutely not a single sunny day. It will be drizzling or it will be cloudy for 3 months solid, and plants will be kind of pampered with this kind of season. So what happens is when, say, in September, there is a suddenly there is a week-long bright sunny day and temperature rises. Suddenly, we can see many plants wilting in our compound. Like they did not wilt during the hot summer, but now they are wilting, even though they were getting such a good rainfall for past 3 months, and suddenly they start wilting. But by September, October, November, there will be intermittent rains. So maybe after a gap of 1 or 2 weeks, again it will start raining and all that. So they somehow manage. And again, from February or like Jan onwards, the frequency of rain decreases, and these plants kind of respond better. So the gap of hotter days increase, and the adaptability of plants also keeps increasing, and they very well survive in the summer. Now they don't wilt. So the same plants which were pampered for 2 months could not withstand a hot temperature for 1 week, but the plants which are not pampered for a few months were now able to survive the summer season of March, April. So this is something which the farmers observe, which even many of you would have observed. And there is a paper which came in 2014 which explains like how these plants or why these plants adapt to stress. So this is just an example of rain and dehydration, rehydration. But there are many other examples also. I'll come into that. So that means plant was able to remember something, right? They were able to remember a stress, and they learn to live in that stress. So at a later time. So that is what we call as priming. That is, the plant is getting ready to face the next stress. But this memory can even go beyond the life cycle of the individual. So it can transfer this information to its progeny and even subsequent generations. And that's what we call as transgenerational memory. So it's such a simple thing which will have its secret hidden in the molecular side, which we call as epigenetic memory. So let me give you some more examples of stress-induced transgenerational phenotypic changes. So there are previous reports where heat stressed it increased fitness in terms of seed production per individual in Arabidopsis plant, and this kind of was existing in the subsequent two generations, so progeny and grand progeny. Even grand progeny to certain extent showed increased fitness. So there are many instances, this fitness level comes down, but still it will be lingering around. So in Brassica, herbivory had induced better plant growth and production, and this particular phenotype existed even in subsequent generations. So these are some of the previous reports. And now, if we talk about a change in the any physiological changes that we see is having its trigger in the genome, right? The DNA will be regulated in such a way that the transcription and translation is affected, and what we see as a phenotype has answer hidden in the translation, right? But now, what is happening in the genome which is affecting transcription and translation? So generally, there could be two types of changes in the genome. One is the permanent change and one is the temporary change. The permanent change is what we have been knowing for quite ages, which is a mutation or change in the genetic sequence or genetic material. And the other change is what recently for past 1 or 2 decades, people are talking about epigenetic inheritance, and past 1 decade, there is a more focus into this kind of inheritance, where there is no change in the genetic sequence as such, but some other modifications happen in the DNA. We'll see that here. So epigenetic changes are something to do with the chromatin. Chromatin, which comprises of both DNA and histones. And the chemical modifications of DNA and histones affect the conformation of chromatin, which in turn affects the gene expression. So these changes are at the regulation level. So these are the routinely seen chemical modifications of DNA and histones. DNA mostly undergoes methylation, that is adenine, cytosine, guanine, thymine, they undergo methylation, and histones can undergo methylation, acetylation, ubiquitination, phosphorylation, and there are even instances of small RNA, non-coding RNA is also involved in affecting the conformation of the chromatin. So basically, when epigenetic changes has something to do with gene regulation at the transcription level. So this is what happens when histones undergo acetylation. So the tightly bound chromatin loosens when there is acetylation, and this in turn increases transcription. So here acetylation loosens chromatin, methylation condenses chromatin, phosphorylation, if it happens near to a methylated amino acid, it can loosen the chromatin. So these modifications
We cannot hear you, Jasmine. I think got disconnected. Yeah, let me call her. Yes, Dr. Jasmine just got disconnected, and please wait for a while. Madam is going to join back.
Okay, sir.
Oh, I'm extremely sorry for the break.
No issues. You can just assume from that slide.
I think I was here. Okay. Yeah, so that's about histone modifications, and here we have DNA cytosine methylation. So methylation or demethylation both affect gene regulation. That is, this is so when a methyl group is already present on the DNA, if it is taken off, that will lead to epigenetic changes, or a additional methyl group. More than one methyl groups can be added onto the DNA. That will also affect gene regulation. So epigenetic changes are all about these kind of chemical modifications on the chromatin, and that they actually do not change the DNA sequence. Well, that's in eukaryotes, methylation of cytosine is more common, and that's what affects gene regulation. And other nucleotides actually are not much prone to such changes. But in prokaryotes, we can see methylation of adenine also. Coming to plants, methylation is in the promoter is mostly in the form of repeats of C and G, CG repeats, which is called a CG methylation, and which is this is common in animals also. In addition to CG methylation, plants also have the CHH kind of methylation, where H is any base except G. So repetition of CHH in the promoter region or any other regulatory region, as well as CHG type of methylation. So symmetric and asymmetric type of methylations are seen in plants. And why is it that methylation affects memory? So that's because methylation is a process which can be inherited after a cell replication, that is from cell to cell, and this replication can be mitosis or meiosis, right? So that's why it can go to the progeny also. So before a mitosis or meiosis, what happens? DNA replicates, And look, you can see into this picture. So this is a bit of DNA which is going to replicate. Some of the nucleotides are methylated. So what happens after replication is, this is the nucleotide which is not methylated here. It just goes unchanged. It doesn't get methylated, and that is faithfully carried over. And look at this nucleotide which is methylated, and this one during the replication, the newly added daughter strands, though do not have the methyl group, soon methyl groups will be added just opposite to the parent strand. So this is the reason why the methylation is carried over after mitosis as well as meiosis. So there are enzymes, methyl transferases, and many other enzymes playing important role in this. I'm not going to confuse you with the names of all those enzymes, but in simple terms, this is what happens. And this is a reversible change. Suppose the organism does not want to remember these changes, it can go back, revert back to its original state also. So if we compare genetic and an epigenetic variation, it is something like this. Over generations, multiple generations, mutations actually accumulate. Genetic variations keep accumulating. They do not revert. There is no reversal. But epigenetic changes are not like that. They do not keep accumulating over generations. It keeps changing up and down. It can go back or go front. So that's the major difference in the trend of genetic and epigenetic inheritance. Both are heritable. So these days, when we talk about genetics, we have to have this as epigenetics also as the other side of the coin, because both are affecting phenotype. So both are dealing with inheritance and variations. Now, stress-induced transgenerational epigenetic changes. So previously I had showed some slides where a stress had caused change in the phenotype. Now, there are recent papers which support why these stresses cause phenotypic changes, and these papers have revealed that many of these stresses cause loci-specific methylation changes as well as global methylation changes. So this is an example of loci-specific methylation changes due to radiation, and here temperature as well as UV radiations have caused in rice as well as Arabidopsis has caused. Loci-specific means they have seen addition or deletion of methyl groups in promoter of certain loci, certain gene loci. And there are reports where people, they were scientists were curious about natural disasters where radiations had were important mutagens like the Chernobyl accident. And it was a survey of that geographical location had revealed that many of these plants there actually survived better or adapted better after the accident. For example, Spinus sylvestris. It was able to survive better in the radioactively polluted soil. And scientists were curious, and the naturally existing Arabidopsis plants were taken for further molecular analysis. And these plants, interestingly, they had a better tolerance to mutagens like rose bengal and methane sulfonate. And the global genome of these plants were hypermethylated. They had more somatic homologous recombination rates, and the gene expression of genes involved in DNA repair and other pathways were altered. So naturally well adapted plants had something to do with their genome and epigenome. There are a good number of papers describing various biotic and abiotic stresses, as you can see here, which have led to changes in histone as well as DNA, methylation of DNA and histones. And this memory has lasted for a few days, as you can see here, or up to progeny, which is called as intergenerational memory, or even grand progeny. So that is transgenerational. So why this terminology intergenerational and transgenerational? Because in animals, germline is not like plants. There the germline is predetermined. That is, an individual who is undergoing a stress will be actually carrying the sperm or the egg meant for the progeny. So a stress which the individual is undergoing could be experienced by the gametes also. So we cannot call it as a true transgenerational. That's why this terminology intergenerational has come to describe epigenetic inheritance of the progeny. And if that goes to the grand progeny, then it is called as a true transgenerational memory. However, in plants, not necessarily that the plant would be in a flowering stage. So it could be intergenerational or transgenerational in plants depending on the situation. But many papers prefer to use demarcate these two terms even in case of plants. So there are some reports revealing what kind of proteins are involved in maintaining epigenetic inheritance. This is especially when a plant encounters a stress. So this particular paper had subjected Arabidopsis plants to very different kinds of abiotic stresses, and these stresses increase global methylation, that is overall methylation of the genome, and also led to enhanced somatic recombinations, that is random recombinations in the genome. And these plants survived well in higher stress, and it was found that two proteins, dicer-like proteins, DCL2 and 3, are involved in this global methylation process, of which dicer-like 2 is required for the memory to go to the progeny. So if DCL2 is not functioning well, then this memory will not be transferred to the next generation. So like this, scientists are in the process of unraveling individual key players in epigenetic inheritance. So now this is about memory inheritance to the next generation, but I would like to remind you that epigenetic changes is not always about memory. Epigenetic changes are even otherwise a part of gene regulation which affect phenotype. For example, in this case, you can see that sequence of tomato, a gene in tomato has undergone epigenetic modification. So here we have the one upper strand is of the phenotype mutant type, or I wouldn't call mutant, an epimutant type, which gives a distorted phenotype in the tomato, and this one is a normal. So in some cases you can see here the white spot, the white boxes indicates demethylation and black ones methylation. So this difference in the methylation status of the sequence and not the sequence per se has led to a phenotypic change. So here epigenetic changes are something to do with the phenotype, but not as a memory, which is due to some stress. This is this is something which is existing in the nature even otherwise. Now, I show you another interesting example where epigenetic changes is required for routine survival of the plant. For example, this in Arabidopsis there is a gene FLC, which is if expresses represses flowering. So expression of FLC gene actually does not permit the plant to enter into the reproductive stage. It is arrested. However, when the plant is subjected to winter season, a cold temperature, this gene now gets epigenetically modified due to the cold stress. Okay, and as a result of which the FLC gene expression is inhibited, which in turn permits flowering. So a cold season is required for the plant to enter into flowering season, and the secret hidden there is in the FLC gene. So this is something, required for in many organisms, especially in plants, if they have to if they have to break their hibernation or enter into reproductive state, this kind of phenomenon is seen. And that means epigenetic change is required for its survival, right, to go to its next generation. So again, this is not something to do with memory. However, epigenetic changes, whether it is for the routine gene regulation or for memory, the interesting point is that both use the same enzymes, same mechanism. Like the methyltransferases and other proteins which are involved, everything is almost same. But what is it that demarcates the trigger or the control which decides, okay, this is the routine epigenetic change for the gene regulation or for the maintenance of the organism, and this is only to be triggered when there is a stress. What is it that decides? This is still to be discovered. So this area is still vague, but yes, it is a fact that epigenetic inheritance is playing a very crucial role in maintaining the phenotype and survival and adaptation of organisms. This is just about plants, but same thing is seen in animals, in humans also. Now, this is all general about epigenetic inheritance and memory. Now, let me come to my story. So,
Yes, Jasmine. It got disconnected in between. No problem. Now I think it's okay. Could you hear? I think got disconnected again. Let's wait for a while.
I am extremely sorry. This is actually because of the bad connection or whatever.
No issues. No problem. Yeah, you can continue.
Actually, we are having some power issues here
Okay.
with the Wi-Fi because there was a very bad wind last night. So there is some power issues going on in this locality. That's a reason. Okay. Yeah. So coming to my area, so I was actually not a hardcore epigenetic person. All these slides which would have given you a misleading feeling that I am into too much of epigenetics. It's not like that. Actually, I started my my research career with Agrobacterium, whether it was PhD or a postdoc. I was always sticking on to Agrobacterium, either to generate genetically modified plants, or then I was more interested in understanding plant agrobacterium interaction, uh, like how the tDNA enters and what proteins are involved in its integration and all that. And in that process, one part of my research was to study whether agrobacterium is affecting the host genome stability, the mutations like point mutations, homologous recombinations, and so on, apart from its tDNA integration. And with that in the backdrop, let me take you to my work. So I think all of you are familiar with this organism Agrobacterium tumefaciens, which is an important genetic engineering tool. I think even school students are familiar with that. But let me remind you that apart from it being a genetic engineering tool, it has unraveled many secrets in biology. For example, even gene silencing, what we know today, was first discovered in plant, and that too when petunia were tried to where scientists were trying to make petunia uh, become more darker, that is from light blue to dark blue, by adding more transgene encoding the chalcones in this using tDNA. So there from like there are many such examples where Agrobacterium tDNA GM modifications have unraveled other secrets like silencing, homologous recombination, and so on. There are many discoveries in the plant agrobacterium interaction area, which have been excited the minds of scientists working in this area, starting from 1942, where first time it was discovered that crown gall could keep on proliferating even in the absence of Agrobacterium. That was the first example of a natural genetic engineering taking place in the organisms. So that was the first example where a foreign tDNA could enter into another organism. And I'm not going into all those details, but let me tell you that scientists were always hunting for finding which protein is the crucial key player during tDNA integration. What happens during tDNA integration? That is, the tDNA enters into the genome, fine, but the host genome paves way for a foreign DNA. How? Whether it breaks and this enters, if it breaks, what are the proteins which allow the breakage and joining and all that. So one protein was discovered, but finally, it was understood that many proteins are taking place, and the key player in this is not yet identified. And these proteins were not mandatory also. They were always plan A, plan B, plan C for doing the same job. So this was kind of confusing scientists. And when we read the literature, you are kind of you kind of feel that what people have done is, like the story of elephant, where many blind men are asked, who have not seen elephant at all in their lifetime, when they are asked to touch the elephant and describe how it is. One guy touches the leg, other guy touches the trunk, and other guy touches the tail, and they start describing elephant in their own uh, language, or in their perspective. So that's what happened here also. People started describing tDNA integration. Everybody was right, but it always had some other thing hidden there. So I guess you're all familiar with these happenings with tDNA entry and integration. I'm not going into that, but what we were trying to understand is who are the key players in this. So my student, now she has completed her PhD, Dr. Joyce. She and me together were working on these lines, and we thought of working on DNA repair mechanism. So most of the previous papers, somehow they were focusing on defense genes. So all sorts of defense genes were analyzed in the presence of Agrobacterium, and in its absence and compared and all that. And it had kind of taken a different route. And we thought of attacking the DNA repair mechanism directly, because that's what happens. DNA is nothing but a DNA. Our genome is a DNA. So something has to join, break and join means it has something to do with repair. So with that background, so that this was actually my inspired topic, DST inspired topic as well. So it's from here that we start. So yeah, and all of us are aware of these different mechanisms which are common in eukaryotes, all eukaryotes, nucleotide excision repair, base excision, homologous recombination, and all that. So these DNA repair mechanisms were selected, and genes from different mechanisms where important key players are selected, and we went on for further planning. So now, what Agrobacterium is made of? Agrobacterium is unique in the sense that it is not just a mere bacteria. Apart from the mere bacteria, we have three additional key players. One is the Vir proteins, which is required for tDNA entry. Other is oncoproteins, which is required for making the crown gall, and other is the tDNA itself. So, if we think Agrobacterium as four components, one the bacteria and other three these things, we can narrow down or expand rather Agrobacterium into different strains, that is, which are different from each other in the presence or absence of these factors, but with the same chromosomal background. So we instead of working with one or two strains at a time, we started working with all these strains at a time, and we went on for looking into expression of all those DNA repair genes which I showed you in the previous slide. And most of the genes, in fact, all the repair pathways which we analyzed had change of gene expression. So every other pathway was affected due to this, due to all of these strains. So that means it's not just one pathway or say it's not just homologous recombination or non-homologous enjoining or like that. It is everything together which has been affected. And we also analyzed them at different time intervals, and we found that 24 hours was giving peak change of expression in all the cases, most of the cases. So we could actually sort them out based on presence and absence of these factors and time intervals and all that. But now the thing is, we can tell that of course all these pathways are involved, and that was one side. That is on the DNA repair gene side. So what is happening to the DNA as such? So we asked the question, well, we cannot see tDNA entering the genome, right? That's not practically possible, but can we get some indirect information? So we started looking out for whole genome strand breaks in the plants when Agrobacterium is encountered. And we looked into the previous papers, we could not find such report. And we started infecting plants at different time intervals. And Joyce was smart enough to pin the time 4 hour where we could see DNA strand breaks happening randomly in the genome. And this was irrespective of the strains used. That means just the bacteria was enough to cause these kind of breakages, but for tDNA, but the expression of the gene was different. So this was irrespective of the bacteria, but this was not irrespective of any different factors, but was specific either to Vir proteins, tDNA, oncoproteins, or just the bacteria. So these are the two different kind of responses that we find. Now the question is, if all these genes are to be expressed, are they controlled or are they regulated by the Agrobacterium? That's when the concept of epigenetics started coming into our minds, and we started looking out for promoter methylations. So I'm not going into this PCR. I think many of you will be knowing methylation sensitive restriction enzyme digestion, and PCR, which is good for looking loci specific methylations, not whole genome, only loci specific. So we use this technique to study the promoter methylation status of all those DNA repair genes which I showed earlier. So here is an example. If a site is methylated, you'll see a band. If it is not methylated, there will not be band. So it is as simple as that. Band is methylated, no band, no methylated. So with that simple explanation, I'm not complicating you with this technique. We went on to study the methylation status of all these genes in the presence of various strains at different time intervals. So here also we took different time intervals and we started, and we found all sorts of changes in methylation, DNA methylation. That is, some genes are demethylated. As you can see here, this control is methylated, but infected 4, 24 and 48 hour infected are not having any band. That means demethylation. In the second example, you can see here, control is not methylated, but one of the infected is methylated. So that also, we found some genes which belong to this category. And here there were no change. Control and infected were the same, no change. So that we found some genes belonging to this category. So like this, we analyzed all the genes. I'm not showing data of all the genes, but yes, we were able to categorize these genes, whether they were showing difference in the presence of tDNA, Vir proteins, oncoproteins, and so on at different time intervals. And to pull entire information together, so tick means methylation, no tick means no methylation. So we could categorize them again into different categories of Vir protein, tDNA, oncogenes, and at different time intervals, of course, to put it in a simplified term, we could actually pin down the signatures, epigenetic signatures induced by different bacterial factors on the promoters of these genes. So with this, it was clear that Agrobacterium has much to do with the genome. It was not just in simple words tDNA integration, but something which is happening at a global genome and epigenome level, which could be because of to some extent controlled by DNA repair genes. Now, obviously, you know, science is like it keeps on triggering curiosity. You get one result, you stumble on to some curiosity, and that takes you to another level, right? So with that, I take you back to this slide where I was talking about transgenerational memory. Obviously, if we see an epigenetic change in the plant, now the question is whether this memory is transgenerational, right? So even a 4-year-old kid may ask this question. So with that, we went ahead in analyzing second generation, which was not infected with Agrobacterium. Only the parent was infected, and again the third generation. Okay, so we took all those genes into consideration, and we found that of these 6 parental generation, it did not go to the next generation. 5 showed memory even in the progeny. That was when, you know, we had excitement of the lifetime. We could pin down precisely what was happening to these genes in their promoters. And interestingly, in the third generation, 3 important key players, okay, belonging to NER pathway, NER is nucleotide excision repair pathway, homologous recombination repair pathway, and even non-homologous end joining. So 3 key players of 3 different pathways showed memory in the third generation. So we could pin down their epigenetic signatures on their promoters. So now what was all this to do with tDNA integration is again another discovery to be made. But these 3 different genes were interestingly or coincidentally affected by different factors. So SEN2 because of Vir proteins, RAT54 because of tDNA, and LIC4 because of oncoproteins. This we were able to tell because we used different strains right in the beginning of our experiment. And another interesting thing that we observed was this epigenetic changes. If we compare across the generations, say here we have generation 1 and 2, 1, 2 and 3, there was a reduction in the level of methylation as well as demethylation. Here you can see the number of bulbs has reduced over generation. In CEN2 you can see it has reduced, even here it has reduced, and so on. So these were some of the observations which we came across at a very, very close level of the promoter. So we are in direct contact with the gene expression and the promoter in terms of DNA methylation. And now we are from the elephant story, we come to this concept where Agrobacterium interaction is like triggering too many things at the same time. Well, it was not, this is not a very unique thing. Okay, let me tell you, don't think that, oh my God, this plant genetic engineering tool, what it is doing to the genome. No, it is a pathogen. First of all, Agrobacterium is nothing but a pathogen. And let me tell you, like Agrobacterium, other organisms also cause epigenetic changes. So here we have previous example of Xanthomonas, which has caused hypomethylation of defense-related genes. Similarly, a virus and another bacteria. So these are examples of loci-specific methylation changes due to defense, which altered defense genes. But ours is an example where it has affected DNA repair genes. But not only DNA repair genes, but it is showing memory also. So this was the first report of biotic stress inducing transgenerational memory on DNA repair system in the literature. So that is what we got published recently, and it has kind of attracted media and all that. I'm not going into those things, but yes, one thing is there. Of lately, for past 1 decade or so, there is a lot of research going on in the area of epigenetics, like how previously, people were more interested in unraveling what genetics is, and then later on understanding the genome sequences and understanding transcription, regulation, all this because of based on the sequence information. And now it is like the trend is entering into what is hidden in the epigenomes of organisms, especially stress-induced epigenetic changes. So with that in mind, I think I should, I'm not going into the next area because I think I have entered my time limit zone. I'm not taking the remaining slides, but yes, I would like to wind up with this slide of mine, and will come to the acknowledgement slide. Yes, so that's my PhD student Joyce, who has done most of the work, Chandani and Swati, who I am actually very happy that, my students had traveled along with me and to take me to this world. So this was the concept. DNA repair genes was the concept which I proposed for my Inspire funding, DST Inspire funding. And when Joyce joined me as my first PhD student, we together entered into the world of epigenetic analysis, and now we are currently in the process of whole epigenome sequencing of plants subjected to Agrobacterium and other microbes. And thankfully, Joyce was so excited with this result that even after she submitted her thesis, she used to come to the lab to infect the plant with Agrobacterium, extract DNA, and give it for whole genome sequencing also. So, whole epigenome sequencing. So that way, this is a very, it has triggered an excitement in our lab. And yeah, I'm thankful to my PhD guide who gave the Agrobacterium strains, and Paul Huikas also, who provided with appropriate Agrobacterium strains without which it wouldn't have been possible to do this kind of analysis. and of course funding my own university, Inspire and Serve also. And with this message, I would like to wind up my presentation. Thank you so much. So your questions are a great welcome.
Yeah, thanks a lot, Jasmine. It's a wonderful presentation. Yeah, I really enjoyed the way that you spoke, and also even the final concluding slide that you said that your own PhD student is you were learning together. You were actually doing into the the new field of epigenetics from the Agrobacterium, in which you specialized for your postdoc as well as PhD. Wonderful work. I really enjoyed the presentation. We are open for any questions from the students or my colleagues. You may please unmute and speak up. Any questions or comments? Jasmine, can you just say a few words about how do you do this analysis, like methylation? How do you actually perform in the lab? So what kind of tools and techniques that you use for assessing the methylation patterns?
Okay. So if we are looking into, say, I will tell about analyzing methylations in DNA. So if we first we need to know what are we looking out for. If we are looking out for whole genome, whole epigenome change, that is methylation in the entire genome, we need to extract the entire DNA subjected to bisulfite sequencing, give it for, we have to give it to an external sequencing agency or whatever and do the analysis. But if we are looking at certain loci, like you want to look at this place. So here we can again go for two methods. One is PCR amplify the region of interest subjected to bisulfite conversion and give it for sequencing. That is one thing which is possible. Or if we want to look at certain regions, because in promoter it is easier to look out for regions which are prone to methylation, because I showed you in one of my previous slides, you will find these repeats of CG or CHH. So such repeats, such regions can be selected, and what the method that we chose was to look into these kind of regions. So there are certain restriction enzymes which are sensitive to methylation, and we took the advantage of these restriction enzymes. So these enzymes are called, like MSRE is nothing but methylation sensitive restriction enzymes. So in normal conditions, these enzymes would cut the DNA, and when we subject this digested DNA to PCR, obviously we will not see any band, right? However, if the DNA is methylated, okay, then these enzymes wouldn't work, and this digested DNA would give a band. So that means methylation is equal to band in the PCR and vice versa. So with this, we took advantage of many restriction enzyme sites present in the promoter region. Not just one, at a time we took many sites, and then we did this analysis. So these are the sites present in each of these gene promoters, and that's how we did the analysis. So whatever table I showed here is a consolidation of all these restriction sites. But a better method than this is giving it for sequencing. I could not give for sequencing because of some simple reason, geographical location of Kasargod. In 2012, those days when I started, we were not well connected to other places where we could send our samples very frequently for sequencing and all that. There were a lot of issues with degradation, and these experiments, we have to do it at a large scale. Like we are doing too many treatments at various time intervals at the same time. So we need to have all the samples sequenced at a stretch. So even if few samples degraded, we had to start everything from the scratch. So because of this problem and the fact that we have to always depend on an external agency for doing this, well, Delhi is a kind of place, means Delhi, Chennai, they're well connected. There, such issues won't be there. But for us, it was easy to stick on to a technique which is doable in the lab itself. That's why we took this technique, which was very reliable. There was no external interference. We could do a large scale experiment at a very narrow level. So suppose you are going for whole genome sequencing, you cannot do too many treatments and too many time intervals, right? Because it would be too expensive. So, and what happens is it would be like you're lost in a huge jungle. So rather than getting lost, we decided to strike at the right at the note, right at the promoter, taking many parameters at the same time so that we can strike the right note. That's why we chose this technique. So we need to decide on a technique depending on our experiment. That's what I want to tell. Depending on our experiment design, what you are looking for, you can look out for technique. Now, this is about techniques used for DNA methylation. Likewise, there are other techniques for histone methylations also, which again, maybe I can explain it later. I don't have the slides for that now. So yeah, chip analysis and all that.
Okay.
So,
Yeah, fantastic. Yes. Very good, Dr. Jasmine. So I just have another question about the practical significance of this work. So there are a lot of talk on climate resilient crops, and climate mitigation. One form is adaptation. So is there any significance of this work in terms of climate resilient crop?
Yes, of course. I had showed you most of the examples that are existing in the literature are based on abiotic stress, and the climate changes are again a abiotic stress for the plants. So what now the concept is more into Lamarckism, that is adaptation, as depending on whether it is climate or any other stress, plants tend to adapt, and the secret hidden is in the epigenetic changes. So we can actually tune the plant for a favorable either climatic condition, drought, or any other situation without changing the genome. So that is the next thing which is going to come. So here we are not going to change. So we will not have the GM. We will have epigenetic change, which is again a reversible, but we will get a desirable phenotype. Now we need to identify what are the factors, triggering factors for a desirable epigenetic change. So say you want to have a particular epigenetic change in such and such region of your genes, which will tune the gene regulation such and such way, and what external factor can do it so that this plant is well adapted to a situation.
So is something like the CRISPR-Cas, is there any site-directed epigenetic changes that we can do as of now? Do we have any technology for that?
That's a wonderful question. I think people in the area of epigenetic are hunting or looking out for something like that. So far, like how CRISPR-Cas works, it's sequence specific. I'm sure soon this will also come.
I see. Very good. We have one question here from Asima. She's asking, plant response to abiotic as well as biotic stress. Is there any common epigenetic response during the interaction of plant with its biotic and abiotic environment?
Oh, yes, there are many overlapping pathways. So it's not just one or two genes. There are many genes involved in the pathways. And if we look into the whole epigenome sequences of plants, we can see common biotic and abiotic pathways. So she can read such papers, and if she's looking out for such precise tags, it will not be impossible.
Okay. Any other questions from any of the participants? Is Dr. Pankaj here?
Hello.
Yeah.
Hello. Good morning, everyone. Ma'am, my question is, that your data shows that after first generation and second or third generation, the level of methylation is decreasing. Even the conditions are same, like the stress conditions are maintained. Even then, the level of methylation is decreasing. Ma'am, why is this methylation decrement is taking place in second and third generation?
Adesh, there is in your question there was a small misunderstanding. We did not give stress to the second and third generation. Stress was given only to the parent. So the parent has transferred memory to the progeny and grand progeny. But suppose if there were, in simpler terms, 5 changes in a particular promoter, of the 5, only 3 has gone to the progeny. Okay, that means of the 5, 3 are required for memory and other 2 are not. So something like that. So it has reduced but not erased completely, possibly because the child was not subjected to stress at all. So child only remembers the stress which was given to the parent or the grandparent. Actually, I will share with you one interesting study of Holocaust survivors. Okay, epigenetic study was done on the current progeny of progeny whose parents or grandparents survived during Holocaust, and epigenetic signatures of trauma are still existing in the current generation. That strong this epigenetic inheritance is even in case of humans. So there are recent reports that bad habits, I wouldn't say bad, it's a relative term. Okay, let me call it as habits like obesity, eating, smoking, all that is causing some kind of epigenetic change in the genome. Some signatures are laid on the genome. Some language is created, and this is existing in subsequent generations in humans. So now these kind of reports are coming up of lately. Many reports are coming up. So we think that what we are doing is, only we know it. No, it's not like that. Our genome knows more of it, and every secret is written there. So it is just the time that we learn to decode these secrets. So in past 4-5 decades, scientists were busy decoding the genome sequence. Now they are going to be busy decoding epigenome sequence.
Excellent. So I just want to share, this is the media coverage of her story. So as we can see that the plants transfer stress experience to DNA repairing gene of offsprings. This has been covered all India by the Indian Express. And of course the local, in the Kerala, the news as well. This is a news from the Mother Bhumi. And thanks so much, Dr. Jasmine. You spent almost more than 1 hour, 1 hour 20 minutes with all of us here. And in case of any questions arising still further, because it's something that you put up for our mind to think, right? Mind will wander with curiosity-driven questions. So in case some additional questions come, I will let you know, right? So I will put in the comment section of Facebook. So
good morning, ma'am.
Yeah, anybody would like to add some point or some comments about today's talk? You're welcome.
Yes, ma'am. Is there any pattern that reflects a balance between enzyme activity that install or maintain or remove the methylation in plant cases?
Sorry, I didn't. Can you repeat?
Is there any pattern that reflects a balance between enzyme activity that install, maintain, or remove methylations throughout the generations of plant?
Pathway which induces methylation? Yes, of course. There are many genes, I told you, right, belonging to the category of methyl transferases, demethylases, and so on. Even dicer-like proteins, some of those involved in small RNA interference also. So these pathways are involved in removing or adding methyl groups to DNA.
All right. Yeah, I hope.
Have I answered your question?
Yes, ma'am, somehow, but I didn't find the answer that why epigenetic changes happens only only throughout the generations of the plants that leads to the methylations and demethylations of
Why it
euchromatin or
happens? You
yeah.
mean to why it happens? Because why it happens is it's a part of gene regulation actually. So this is how plants have evolved, or even not only plants, eukaryotes have evolved because a organism cannot afford to cause genetic changes, right? Because a situation may be temporary. So spontaneous mutations have always been a part of evolution, which have allowed organisms to survive better or lose the battle, but not necessary that every change need to be permanent. So there, the evolution has found out some mechanism for a reversible change. So that's where epigenetic comes into the scene, where something which is reversible yet will tune the DNA will behave just like a genetic change, but if the organism wants, it can go back to the original form. So it's probably a part of evolution, a mechanism which life has discovered for itself.
Okay, we have one more question here from Mohammed Singh. Mohammed Singh, could you please unmute and ask the question directly?
Hello, ma'am. My question is how this all changing in rain cycle and this the climate change, how to what extent it's affecting the epigenetics of the plants?
Yeah, it is affecting to a very, very great extent. There are many studies, reports in where people have analyzed seasonal variations. And yeah, I think your question is similar to what Dr. Felix had asked before. So yeah, there are many reports and whole epigenomes are sequenced and studied. Even naturally existing trees, and they are also being studied. To understand how climate has affected. Yeah, the thing is, currently people are doing all this whole epigenome sequencing and telling, yeah, this has changed, that has changed, all that. But still, we are yet to understand what does a change mean. So, say there are the different pattern for particular climatic change. Yes, that the whole epigenomes are sequenced, but it is like too much of research is required to at least understand what this change mean. So it is something like, we started 40 years before understanding whole genomes. So people used to sequence genome, and then try to understand what each and every sequence means, what are the regulatory regions, what are the non-coding regions, what do they mean. It's a similar situation. Now again, we are trying to understand what these means and how climate, yes, climate is causing change, that is a fact, but what is it that it wants to convey is much to be understood here on.
All right, so thanks a lot, Dr. Jasmine. It's wonderful to spend time with you all, and it was really helpful for us all to increase our understanding about what epigenetics is and various molecular ways, and how do you study the epigenetics and what are its significance. And yeah, my best wishes for your group, especially your PhD student. I think she has completed her PhD, right? I think she has submitted the thesis, and yeah, I mean, you said that she's still coming to the lab and doing the work. Yeah, that shows the internal driver for doing something interesting, right? So yeah, I mean, I'm really happy to know that. My best wishes for your group, and please do more enlightening, more inspiring, and curiosity-driven research like this. I hope you get many more papers to come to your lab. Yeah, my best wishes to your team, and yeah, have a good day. Thank you for coming.
Thank you, Felix. Thank you.